So we'll get the ball rolling here. So hello, everyone. I'm Brett Weeks. I'm the Agricultural and Environmental Coordinator of Lamont County, and I will be moderating the event today, along with Hannah Muster, the Agricultural Service Technician from Vermilion County, who will be running some things in the background and organizing your guys' questions. It is great to have so many of you logged on here today for this event, and we're happy to have you all. So I'd like to just say that this event is brought to you from the County of Vermilion River, the County of Lac-La-Biche, the County of St. Paul, the County of Provost, the County of Two Hills, and the County of Lamont through the Clubroot Surveillance Program. I would like to take a second to mention that this program was funded for by the Government of Canada, the Government of Alberta, through the Canadian Agricultural Partnership. So getting started into our Clubroot 101 online workshop, we have 4 speakers here today that will hit into topics about Clubroot management, prevention, pathotypes, genetic management, as well as some other areas. Our speaker order for today will be Aaron Van Beers from Leduc County, then Leighton Blaschko from BASF, followed by Dr. Steven Strelkov from the U of A, and we'll finish off with Dan Stanton from Corteva. There will be a time at the end to ask questions to our speakers individually after their presentation, and then if you missed that time frame, hopefully we can ask some additional questions at the very end of the presentations here. So just to start off with, I'm just going to show a quick video introducing our Clubroot surveillance program and what the partnering municipalities here are doing to manage and learn more about Clubroot. Then we will introduce our first speaker. So I'm just going to link a video here.
Agriculture service boards have been around in Alberta for over 75 years, and as part of our role, we monitor for pests in our municipalities and regions. And one of those pests that we're concerned about is the presence of clubroot within Alberta. Clubroot is a soil-borne disease that impacts canola and other cole crops. It forms galls on the roots, and it can prevent root nutrient uptake, and it can cause stunting and wilting and can cause up to 100% yield loss in some cases. It is easily transferred between field to field if you have infected soil. So that means that the resting spores from infected plants can be transferred into different soil. And if that's spread around, then you can easily spread the clubroot from field to field. This program is the Enhanced Clubroot Surveillance Program, and it's a partnership between County of Vermilion River, the MD of Provost, Lamont County, County of Two Hills, County of St. Paul, and County of Lac-La-Biche. So we're conducting this Enhanced Clubroot Surveillance Program in the North East region of Alberta because most clubroot has been found in the Edmonton region, and we're trying to get ahead of that in the North East region. So we're trying to get an understanding of what kind of pathotypes of clubroot we have here, and then we can share that information with our producers and agronomists in the area so that we can appropriately select clubroot resistant varieties in our North East region. As part of our project, we also do spore collecting as well. So that means that we'll have our little spore trap out in between seeding and a harvest, and we'll try to collect some of the dust movement that's coming off of the clubroot infected fields. And we'll sample that, and then we send it off to the lab to be tested. So with our spore collector, we sample the spore movement for 4 weeks, and so it'll roughly equal out to 120 samples across our 6 counties and MDs that are involved per season. So we have a lot of samples that we're trying to discover the presence of clubroot moving through wind and dust movement. As part of our actual clubroot surveys is that we pull the plants and physically inspect for clubroot galls. So then we check for the presence, and we can see here it's not present in this plant. But that's one of the main part of clubroot surveying is actually pulling the physical plants and checking for the spores. But then as part of our regional project, we're going over and above by using our spore collector and then also testing the pathotypes for when we find a positive plant.
I think it is really good that the municipality is doing this and researching and trying to figure out how to slow down clubroot, because eventually everyone, or most everyone, will have it, and then it's awesome to have the information for when everyone has it and how to deal with it and what we can do, and more information is better.
So we had conducted, I believe it was over 360 field samples. We had, I think, 2 false positives come back, and it was in the paper, and we had farmers instantly terrified, rightfully so. So that's why we've now implemented a new protocol that if we have any suspicion, we send those samples to 2 labs for testing just to be sure. And I know that definitely woke our farmers up a little bit. We have had more people making sure that they're taking proper precautions and being, they're taking things more seriously now. Another thing we're doing is every time we're doing road construction or we're building a new road, it's part of our reclamation program that we take soil samples along that entire road for every field, just to make sure that we're not spreading it. We're very fortunate. I know it's coming, but that's why we do things like this and participate in programs and grants like this, because our farmers, we need to give them the best shot that we can at getting ahead of this. So being proactive instead of reactive is the best that we can do.
This is an innovative program, and the partners are really proud to offer this new technology on monitoring the presence of Clubroot, and we're really excited to share that information with our producers and monitor the movement of Clubroot through the Northeast region of Alberta. So this project has been made possible through the Government of Canada and through the Government of Alberta through the Canadian Agriculture Partnership.
Okay, so now we'll segue right off from that video there that kind of introduced our program right into our speakers. So our first speaker today is Aaron Beers, who is a Southern Alberta farm kid who loves playing in the dirt. He currently works with Leduc County as the Manager of Agricultural Services. He has been primarily responsible for Clubroot Inspection Program in Leduc County since 2009. He spends his summers in Devon with his wife and 3 kids, and his winters in various hockey rinks around the province. And now I'll let Aaron take it over from here. Thanks.
Thanks, Brett. And thanks for inviting me out for this this little webinar. I'm just going to share my screen right now, see if we can get this to work. There. Hannah, Brett, can you see the
looks
Clubroot
great.
101?
Yep.
Perfect. All right. We're cooking now. All right. So yeah, as was stated, my name is Aaron Van Beers. I'm with Leduc County. I've been with Leduc County since 2009, and probably one of the main roles that I've had with my job has been the Clubroot inspection program. There we go. So I was kind of asked just to come in and just talk a little bit about what Leduc County is doing in terms of Clubroot. Talk a little bit about kind of, why are municipalities involved with Clubroot, and why does it seem like every municipality is a little bit different from its neighbor, and then just kind of go briefly into kind of how are we dealing with Clubroot, and how can Clubroot be kind of managed. I won't get too deep into that because I know the presenters coming after me have got some very good experience and probably better knowledge than me on a lot of this stuff, so I'll leave that to them. But first off, why Leduc County? Why is there so much Clubroot in that area, kind of around Edmonton? And I think the former president, President Trump, had a great quote when he was talking about COVID. When somebody was asking him about COVID, why there was so much COVID in the States, he said, "Well, it's because we're testing a lot, and if we weren't testing, we wouldn't have any Clubroot, or sorry, not Clubroot, COVID." And I think that's pretty much the case with Clubroot as well. If you aren't looking for it, you're not going to have it because you don't know it's there or not. But active surveying will show a lot. But I think that's the first step in making sure you know what you're dealing with and what you can go forward to manage. Just to give Leduc County's inspection process just a little bit of a general overview. We, since 2007, have been inspecting every canola field within our municipality. So that works out to be about between 650 and 900 fields a year. When we first started doing this, it was a conscious effort that was put forward to say, you know what, we need to be inspecting every field so that we are we get a better handle on what's going on, and we aren't picking areas or being random about it. We want to make sure we know across the board. Our inspections usually take place that first week in July, basically after Canada Day, once everything's blooming and bolting. That's when we start our inspections, and we go right until the end of September, usually a little bit into October as well. We also inspect any fields that have a large amount of volunteer canola in them as well, because that can be a host for clubroot in non-canola years. So just for an example, in 2020, we did have 647 fields of canola. In our inspections, we did find 150 of them had clubroot. That's probably our smallest amount of canola that we've had in the last 11 years since I've been here. The spring weather was definitely a detriment for a lot of cropping. We had a lot of fields that never even got planted this year. So just to give you all kind of a little bit of an idea of where we sit as a municipality for the number of fields, this is a list. This shows all the fields that Leduc County employees have confirmed have clubroot on them. You can see there's a high concentration within the kind of that between Leduc, Kalmar, and Devon, and that triangle. That's where our best soils are. So that's where we see the most canola being grown, which is why we see the most clubroot kind of showing up in there. And when you look at it like that, it looks like a lot of fields. But when we look at the grand scheme, since 2007, we have inspected 10,500 fields for clubroot. We have had 760 that have been confirmed positive, but that's that number takes into account fields that we knew had clubroot on. They had a rotation. They went back in with canola. We found clubroot again. So we have in our municipality, we have 1,000 fields, just over 1,000 fields that are individual fields that we know have clubroot on them. So, and then another thing, we'll kind of get into how our inspection process goes a little later, but basically 3/4 of our fields, when we do our inspection, that initial inspection comes back at less than 10 plants having galls out of 100 plants polled. So this question kind of comes up quite a bit. Why are municipalities involved with clubroot? What is it that is allowing municipalities to get involved in what is an agronomic issue? And the answer to that is the Agricultural Pests Act. So the Agricultural Pest Act is provincial legislation that is there to assist the ag industry in limiting a pest. So the province, through the Ministry of Ag, can declare an organism a pest. Some of the things that you might be familiar with, rats. Norway rat in Alberta is a declared pest under the Agricultural Pest Act. Grasshoppers, golden nematode, and our good friend clubroot. They're all pests under the Agricultural Pest Act. And this legislation says that if there is a declared pest, a municipality has the responsibility to take active measures to prevent the establishment of or to control or destroy that pest. Active measures is a very broad term. It's actually says that active measures in there. And really the reason for that is that it allows each municipality to kind of tailor a program that it feels will work for its area. So they can develop a program or a policy that works for it. In some circumstances, it can be making sure that fields get inspected. It can be education and awareness, something like this Clubroot 101 workshop that's being hosted by the various municipalities. And it can also have enforcement situations with that. So that's where you hear about a notice being issued when there is clubroot. But really, it is up to each municipality to decide how and what kind of active measures will be included in that policy. So what does Leduc County do for active measures? Probably one of the big things is our inspection program. We do inspect every canola field for clubroot. Each field, we pull 100 plants in a diamond pattern. So we'll go into more or less the approach in every field. We'll walk the diamond pattern. We'll pull 100 plants and we'll look at those plants and see, is there galling showing? If there is what we feel is clubroot, we will take samples and we will send them to the lab to get the plants tested to confirm whether or not there is clubroot there. If we also keep track of how many plants have galls. So if there's less than 20 plants out of 100 that have galls, we make a note of that. We send a letter to the landowner and the renter if we know, stating that, you know, during our inspection process, we did encounter clubroot on this field. It was confirmed. Here's the information. This is what you can do to kind of mitigate your risks. No notice is issued at that time. It's basically an information letter saying that there's an issue. Here's anything you need. Let's talk about it. However, if we do go into that field and we pull more than 20 plants out of 100 and they have galls, that's where we start to wonder, okay, is there an issue here? Is it a high level of infestation in this field, or is there potentially a pathotype shift that's happening? So what we do in those is we send a letter as well, similar letter saying that we found clubroot, but we also inform that landowner and the renter that we're going to be coming out to do a second inspection in the fall once swathing and harvest is done, to try and get a better feel across that whole field. Is it an isolated incident where there's just a small patch by the entrance or where we found it, or is it something that's a little more serious where it is across that whole quarter in high levels? So if it is across that quarter in high levels, we will issue a notice. For us, a notice restricts the growing of canola for 3 years. So 3 years without canola, the 4th year you can go back in with a resistant variety. When we do come back with these secondary inspections, we'll also take a sample. That sample is sent to the University of Alberta for pathotype testing. When we see, especially in a field where we know it is a resistant variety, we have some concerns when we see high levels of clubroot infection. So that is something that we'll send away because we want to make sure that we aren't causing any risks or having any pathotype shifts that we can't deal with. If there is a pathotype shift where there isn't current resistance available, commercially available, we will issue a notice. That notice is similar. We say 3 years without canola, then we reassess in that 4th year. In that 4th year, is there a resistant variety available that works on that pathotype? If yes, then we will lift the notice. And if not, we will continue that notice for another year. Couple other things that we do that I feel are pretty important is we do have an annual clubroot meeting. We hold it. It's an opportunity for us to get that information out to our municipality, to our producers, to say, this is what we found, this is what we've been working on, this is the issues that we're seeing. And then a big thing too for us is focus groups. Every year we bring our policy forward to our Ag Service Board and our council for recommended changes. We try to make sure that we're staying as current as possible with a policy. So we have these focus groups with our landowners, with our farmers, with our retail operations within our area to get everybody's input on it. Because for us, if we're going to deal with clubroot, we need to have a overall buy-in from everybody to make sure that we're doing everything we can, and that to help make sure that clubroot doesn't become a bigger issue than it already is. Just for an example, this is what one of our pathotype notices would kind of look at. We specify the things that we require to be done. In this particular example, if it is an actual pathotype, for example, pathotype 3A, we will identify it in this notice, and then we do say no canola can be seeded, and for the years that it cannot be seeded, and then we talk about a variety that has pathotype 3A available. You can grow that after. So we wanted to talk just real briefly on each of these. How do we deal with clubroot? And there's no real one way that we can deal with clubroot, but I think it is this diagram kind of shows it better than a lot. It's a combination of everything. When we do our scouting, what kind of crop rotation do we have? Our resistance, are we using resistant varieties? Are we minimizing our soil transfer, and are we controlling any weeds or volunteer canola in those areas? Any one thing by itself isn't really going to solve the problem, but a combination of more than one of these can definitely help reduce the risk of clubroot. So in terms of reducing soil movement, this is one that we don't see a lot of activity in our municipality on, especially when it comes to washing or knocking dirt off of the equipment. Went back in 2008, 2009, when we were first starting to have some issues, there were a few producers that did get pretty heavy into this. They were washing, they were bleaching, but the time investment was a little too much for what they were doing. So they've switched, not necessarily washing, but they do knock off most of the visible dirt, any big dirt lumps, as much as they can. And probably one of the key things too is for reducing soil movement is if you have a field that is infected with clubroot, putting that field at the end of the order. So that's the last field that you seed in the year. That's the last field you spray. That's the last field you swath. That's the last field you combine. That way you are minimizing the spread to your fields where you maybe don't have clubroot or you don't know you have clubroot. And it's another way, it's less time intensive to plan it that way, but it's definitely an effective option. Controlling host plants, making sure that there are no host plants in a field when there isn't a canola crop is a good way to help break the spore cycle. So shepherd's purse, stinkweed, wild mustard, any of those types of weeds are susceptible to clubroot. So if you've got a wheat field and you've got a pretty heavy pile of volunteer canola and stinkweed in it, making sure you use the appropriate herbicide to get rid of those weeds is going to be a huge step to breaking that cycle and not letting the spore load build up. Scouting and monitoring for clubroot. Physically pulling plants anytime you're in the field is honestly one of the easiest things you can do. Whether you know you have clubroot or you don't. Anytime you're out in the swather, you got to take a quick stretch break. Just stepping out and pulling 5-10 plants wherever you are in the field is a huge benefit to you, knowing where and when you might have clubroot. Anytime you're going out to scout for what herbicides you're going to spray or what fungicides you're going to spray, and you're scouting for timing, pulling plants and looking for galls is the easiest thing. It honestly is. And pulling from a variety of areas. Although clubroot is likeliest in the entrances of the fields, where you mainly come in with your equipment and leave. One of the interesting things that happened last year was there was Farming Smarter and Alberta Ag and Forestry got together to do basically an experiment to see, can dogs be trained to detect clubroot? So there was a fellow out of Nova Scotia that is a dog trainer. They train for drug sniffing dogs and all kinds of good stuff with dogs. They trained the dogs to detect clubroot spores in the lab. And they were doing pretty good, but they needed to see, does this work in the field? So they actually came out to Alberta last September to come look at a bunch of fields. Leduc County had a couple of fields that we had some producers that were definitely interested in having this happen. And what we did find was that, yeah, the dogs can detect clubroot. It's a lot of money to train a dog properly and to get them to do it, but there is a possibility there. Definitely, it's cheaper to have a seasonal staff or a summer student go do the fields than the dog itself, but the dog might be a little more fun to do it in. Crop rotation, probably one of the biggest things that we see in our region is when there is a better than a 1 and 2 year rotation, we see better results for the management of clubroot. At minimum, a 1 and 3 year rotation can be helpful when reducing clubroot spore load if, and this is a big if the spore load is low to begin with. If you catch it when it's early before you have a big issue, good crop rotation, minimizing your weeds can help keep your spore load down. You'll probably still have some clubroot, but it won't be as detrimental. However, when you have a high spore load already, crop rotation alone is not going to do it. You need to be working in conjunction, and that's where the scouting and monitoring comes in really handy. The sooner you identify that you have clubroot in your area or in your field, the sooner you can implement a strategy and keep it at a minimum. We have many producers in our area that have been growing canola since 2011. They know we've had clubroot in those fields. They've stuck to a 1 and 3, or in a lot of cases, a 1 and 4. And clubroot is still there. Every time they grow canola, we usually find some clubroot, but it's at a low level, and they are managing it. And I think that's a key consideration. And kind of that last piece in the managing of clubroot is resistant varieties. This picture is a very stark example of the good things that resistant varieties can do. This was a kind of a side-by-side comparison that was more or less accidental, but this was from 2011. The field on the left was a resistant variety, and you can see, good swaths, lots of good stubble. This came off quite well. I think it was mid-50s. The right side here was a field that did not have resistant variety in it. Same guy seeded within a day of each other, and you can see that little bit of a swath there. This was less than 5 bushels an acre. So the resistance does work, and it can work really, really well. The issue is going forward is making sure that we have the right resistance for the right fields. This was pulled right from Canola Council of Canada's website. This is all the current clubroot resistant varieties as of October of 2020. The ones with the asterisks beside them are varieties that have kind of additional genetics over the first generation ones. So those are ones that we in Leduc County are starting to see more of, is these varieties being used here. We're kind of on the 3rd, 4th cycle of clubroot, or sorry, of clubroot resistant varieties. So we have to make sure that we know what we have in the soil so that we can use the right varieties and make sure that we can maintain those low spore loads. And then just kind of in conclusion here, can we stop clubroot? I don't know that we can stop it. I think in the video, it kind of, there was a producer there that had said, we're doing everything we can to slow down the spread. And I think that's probably the key message. Maybe you don't have it right now, and that's great. And if you implement a lot of these steps, maybe you won't get it. But if you are implementing these steps and you do find you have it, chances are you found it at a low level, and chances are you can maintain that low level. You won't get to be a big issue where you're going to have to lose that field to canola, to the use of canola. So implementing this as a kind of a whole group is definitely a way to go. So I don't know, I might have gone a touch long there, Hannah, Brett, but if there are any questions, I'm more than happy to answer some.
Yeah, for sure. There was two in the chat box, but I'll just mention two. So the gentleman in that video, Cody, he is one of our producers here in the County of Vermilion, and he hosted our sport trap collector this year. So he was really excited to kind of look into new technologies of monitoring Clubroot, and he's a really good producer to work with. So yeah, we can't thank him enough. So that's really good. But some of our questions that came in, we have one from Laura about how many inspectors do you utilize for these inspections? So in Leduc.
Yeah, so
I would like to know that too.
for our program here, I have one seasonal staff person that is dedicated to Clubroot inspection. We hire them in May, and then this year we were actually lucky. We had them on for 9 months, so that actually worked out really good, because what we find is we don't quite get it all done come the end of August. There's usually inspections in September and October that have to be looked after. So having one, I tell you, I've done it. If you're doing 1 or 2 fields a day, it's not bad, but usually to make sure we get through all those fields, it's like 15 to 20 to 25 fields in a day we have to get done. So it's there's no rest. You got to go and you got to pull a lot of plants. I usually end up pulling, I'd say myself, probably about 50 to 100 fields, depending on the circumstances.
There we go. And then there was also another one coming from Justine. Are you counting, in quotations, fields as individual quarters, Aaron, or grouping them together as they are farmed with multiple quarters or sections together?
Yeah, so each field is considered separate. So there are a couple situations where we have, like a quarter section split into 80s. If they are, naturally separated with a creek or a brush or something like that, or they are farmed individually, we will count each of those individually as a separate field, quote unquote. But typically, these are individual quarters.
And another question here from Tom. Does the County of Leduc fund this, or is this government funded?
So this is funded by the County of Leduc. So every municipality does get a ASB grant. So the Agricultural Service Board grant is a provincial grant that is used to support most of the projects that the ag services departments of each municipality has to do, whether that's weed inspection, pest inspection, vegetation management around roadways and stuff like that. But this is funding that Leduc County does put aside every year for this particular inspection. This was identified back in 2007 that it was important for the county to be involved and to do inspections. So back then we had our council that fully supported it. They put the funds in there for this, and we've continued to bring it forward to our council every year, and every year they do support it. Yeah.
There we go. Well, that's all the questions that are coming up in the chat box right now for you, Aaron. But if there's any other questions that you guys want to ask specifically to Aaron, there will be a little bit of time at the end of our presentation. So I'll toss it back over to Brett. Thank you, Aaron.
Thank you.
Sure, yeah. So thank you, Aaron, for what municipal involvement is for controlling Clubroot. And yeah, again, if you have any more questions for him, we'll try and get them in at the end here. So now we're going to transition into our next speaker, Leighton Blaschko, who is a senior tech service specialist, where he provides agronomic training and sales support for seed treatment, herbicides, fungicides, and InVigor canola. In addition to this, he works with farmers on a large scale, replicated demonstration plots with innovative crop protection products and InVigor canola traits. He was raised in North East Alberta, where he's still active with the Farmley Grain Farm. He's educated with degrees in agriculture, specializing in agronomy from the University of Alberta. He also holds his professional agrologist and CCSC designations. Leighton enjoys photography, cooking, and is a PADI certified driver. So I will let Leighton take it from here. And yeah.
All right, thanks so much. I guess we'll get going here. Let's see if I can share this screen.
Yeah, there you go. That looks good.
Okay, well, thanks so much for the introduction. And yeah, I'm happy to be involved in this webinar. I've worked with Invigor, like you mentioned, for probably about over 15 years now, and also farm in the Andrew area, so in Lamont County. So I'm going to get started, and probably in the interest of time, it's good that some of my stuff is going to be similar to what Aaron had presented. We can just advance these slides. Okay, there we are. So at the end of the day, I think the best offense is a strong defense. So Clubroot can cause major yield loss. Taking an integrated pest management approach is going to be paramount to handling this problem. And then using a hybrid with built-in resistance is just one important tool in that strategy. So from a BASF standpoint, or the Invigor camp, we feel that you can maximize and protect the provincial of your crop with hybrids from Invigor hybrid canola that contain the C within their name. So our naming system would have a C in the naming to make it simple for producers or agronomists to make the right choices for which hybrids have have Clubroot resistance. So I want to acknowledge a little bit of the information that I came from here, and I also put out a bit of a shout out to the Canola Council. They had a really good video. If you Google or search for Clubroot management video, it's an excellent video. It's it's a good watch. And then also there was a international Clubroot workshop here, and there's 10 top tips from that workshop, and I tried to include some of those in my presentation. So these best management practices, you'll see this is fairly similar to what Aaron said. There might just be a couple little ones here, but we say at BASF that sustainability of the canola industry is the most important consideration, and that's what drives our position as to how to best manage Clubroot and how we're deploying Clubroot resistant genetics. So one of the first things in this stop sign approach would be crop rotation, of course. Similar to what Aaron said, allow at least 2 years between host crops. People are trying to grow, canola has been the cash crop, but I think a lot of producers are doing a lot of good things, including more other crops with prices increasing. I think the returns on things like barley and oats have really increased over the last couple of years in this area. More pulses like peas and even faba beans are also having a fit. So I think, that's allowing producers the ability to have at least 2 years between host crops. And you want to reduce selection pressure for Clubroot pathotypes. So you want to deploy resistant hybrids as soon as Clubroot is in the area. I think Aaron referenced this earlier. You don't want to wait until you say, well, I've got a bit of Clubroot. It's really a small amount. It's just in my driveway or the approach. So maybe I'll go another year without Clubroot genetics. You want to make sure that if you've seen even the smallest amount, you want to deploy resistant hybrids as soon as it's maybe not even in your field, but if it's in your area. So I think that's pretty important for people to realize that that's when you do it. You don't wait until it's it's too late, because it's a numbers game. As far as the numbers game, I talk about scouting just like Aaron talked about scouting. And one thing that one of the former Canola Council of Canada agronomists told me is, and I was kind of surprised by this. I've worked in this area for quite a while. And of course, we know that field entrances are low areas because it kind of follows some water sometimes, as well as random areas are areas that you should look for Clubroot in. And you want to pull stubble or plants. So if you've swathed it already, you can still pull at that point. But if you haven't cut your canola and you want to get a little jump, you want to look at plants. And the surprising thing that I was mentioning is that we would say 100 or more plants per field. Sometimes people go in a field and they pull 2 or 3 plants from get out of the pickup and pull 2 or 3 plants and figure, well, that's good. I didn't find any Clubroot, I'm good. But, we were in a situation in northeastern Alberta where we knew there was some resistance breakdown. We went to another one of the farmers' fields and he said, no, I didn't see any there. Didn't see any Clubroot. When we started pulling, we probably pulled 100 plants each. And once we were pulling, in that 30 to 50 to 70 range, that's when each of us found a plant. So you do want to find it if it's there. putting your head in the sand isn't really going to be an effective strategy. So certainly scouting and scouting a lot is, this 100 or more plants would be key in my mind. One more thing that came out of the International Clubroot workshop was about patch management of Clubroot. When you first hear this, you might think as a farmer, are you crazy? What's this guy from BASF talking about? What it would mean is hand pulling infected plants and perhaps grassing that area, seeding grass to 2 times the size of that area. And when you do that, you want to make sure that you pull them out, and then you also properly dispose of them. So, put them in garbage bags to go to the sanitary landfill, or even burning them. I know it's hard to burn green material sometimes, but if you get a good enough fire going and they're dry enough, that's something you can do. The best time to pull this is before those galls start to rot, because those galls, when they're fresh, they're harder. But then once they start to decay, they become more like a sawdust or rotten consistency. So you would have less effectiveness trying to pull them at that point. Now, why would we say that? Why would we suggest this patch management? Again, back to, is this guy crazy? Well, it really can be. I've been in fields where we're doing trials and the farmer didn't know that they had clubroot in a field. And then lo and behold, that's the first year we end up, because we're in these fields looking at our hybrids, doing measurements of height and lodging and maturity and all this kind of stuff, and all these variety trials for InVigor. But then, so we're there quite often, and we're walking a lot of square feet or a lot of those acres where those trials might be. So we do see things that stick out, maybe the size of a tabletop or a pickup truck, or even a room in a house, or even if it was the size of a house. That sounds like it's a daunting task, but if you can pull the plants from that area and pull them, if they're pulling out still with the roots and you're getting those galls out from that one and only patch that's in the area, you will make a difference. The reason you'll make a difference is because each infected plant can have about 16 billion, with a B, 16 billion spores. So that's why this does, even though it sounds like a crazy idea, I think if you do have the very first patch that's very small, it's probably worth, getting some kids out there, a 4-H club, something like that, getting them involved to try to reduce that spore load. Now, those 16 billion spores on each root, you might think, well, they're going to be one pathotype. Well, it's quite different, actually. They're primarily one pathotype, but they're always a mixed population. So they're not always, you can't just say, I've heard so many farmers say, I've got pathotype 3A. And I know right then and there that that's not a completely true statement. They might have 3A, but they definitely have other pathotypes as well, because there are mixed populations in all galls. Another strategy is to consider liming. Now, this also is a bit of out there thinking. It's not very common. It's quite can be quite costly. It's labor intensive. Delivery or freight can be quite a bit, but depending on where your sources are, people are looking at whether this can be something that can be done effectively. So, of course, there's positives from a clubroot standpoint, but the negatives on liming could be nutrient availability limitations. So I'm just mentioning that this is a possibility that's being looked into, and it is somewhat promising. So, that's something that we may want to consider for areas or maybe patches in the future. And then, like Aaron said, control your weeds and volunteer canola as well, not only in the canola crop, but also in other crops in your rotation. So volunteer management is very important, doing a proper pre-seed burn down, maybe changing your herbicide tolerance system up here and there for your canola is a good strategy. Now, slowing the spread through sanitation, Aaron mentioned about, keeping boots and knocking stuff off, their soil off equipment. So consider for your farm having to have a biosecurity protocol. The best sanitation product is bleach in a 2% solution. So it's really important that it would have to be at least a 2% solution. But then also recent research has found that spray-nine has similar activity to bleach. So, maybe that's something that a foot bath, if you're going to have a tour to your farm, or if you're going to have people there, if you can't have the booties or can't be washing footwear or changing footwear, spray 9 in the bleach to make sure that you're doing spray 9 or in a bath, in a foot bath might be an effective way, and it's not as deleterious to clothing and all the staining. So there's a bunch of good resources. Clubroot.ca is one of them. And I think, go there because it's always getting updated. And that's where there's great that we have one, source of where all the information, Canola Encyclopedia, is there about Clubroot. Now, as far as InVigor, how do we position InVigor hybrids? Well, all InVigor Clubroot resistant hybrids have been developed to be resistant to the most predominant pathotypes found at the time of registration. So remember that I said that each gall or each plant that has Clubroot can have different pathotypes. We will be testing them, from as we evolve new products, we continue to test them, but the pathogens are evolving in their concentration, how many there are, where they are. So we test them against whatever's in the soil. We're not introducing pathotypes of everything into these plots because we don't want to introduce them into farmers' fields. So we test them against what they're in the field at the time of registration. So we have a number of first generation Clubroot resistant hybrids, and I'll list them there. I'd be probably starting near the bottom. L241C was one of our earlier ones, as well as InVigor L255PC, InVigor Health L258 HPC. We then brought out InVigor L352C and InVigor Choice LR344PC. So InVigor Choice being meaning that it's resistant to Liberty and Roundup. It's a very small part of our program, but there are some producers that feel that that's an option for their farm. It's a true flex on the Roundup side. And then the latest two that we brought out were InVigor L345PC, and then new for this year would be InVigor L340PC. So that's the list of everything that has first generation Clubroot. And then from our second generation Clubroot resistant hybrids, we have InVigor L234PC. It has first generation genetics as well as second generation genetics. And probably I'm getting to one of my last couple of slides here, and I just wanted to, we tried to do a bit of a flow chart. Now I apologize, this might be a little bit, hopefully the font isn't too small, but if you look on our BASF site for Clubroot information, you can see that we have this same flow chart. We tried to kind of cut through the clutter because to some people, it's difficult to decide. So we wanted to make a decision tree that helps you decide what hybrid you should grow on your farm. So it starts out with, have you identified Clubroot on your farm? Yes and no, yes or no. So if you're on the no side, well, is Clubroot a growing concern for you? If you still say no, well, then you want to grow the InVigor canola that's best suited for your area. That's getting fewer and fewer and fewer. For most people, it is a concern, so the answer yes. And then if you do that, has Clubroot been identified in your farming community? So that's been some language that has been identified by the Canola Council, this word community, because it doesn't mean the community where you pick up your mail. It might mean there's some farmers that are spread across 4-hour drive or 2-hour drive or a long distance. So what is your community? Maybe you bring in a custom applicator from 2 hours away because he's a friend or cousin or someone is handy that you trust for that job. So that your community would be larger than if you were doing all your own equipment is only on your field. You don't have custom application. You only farm within a few miles of the home place. So that's kind of how you would identify a community. So if you said yes, it's in your community, then you want to know, are first generation Clubroot resistant hybrids still effective? If they are, then consider growing a first generation hybrid again. But if the answer is no, then you want to consider a second generation hybrid. The other way of looking at it is if you've identified Clubroot in your farm and you've been growing Clubroot resistant hybrids, so you answered yes on the top left, then you also want to know, are first generation hybrids still effective? Yes or no? If it's no, then you definitely need to be growing a second generation hybrid. If it's yes, we want to ask the question, how many times have you grown first generation Clubroot hybrids? If it's 2 or more, you should be considering switching to a second generation Clubroot hybrid because we could have potential breakdown of the genetics, and it does happen that quickly. If it's less than 2, then you say, is there any symptomology? Well, if there's no symptomology, then you can probably go back to the first generation for that year's choice. So I'm hoping that this flow chart kind of answers the scenarios that you might have for what hybrid you should deploy when. And I think that is all I had. So are there any questions for me?
Thank you there, Leighton. We don't have any questions rolled into the chat yet, but
Okay.
if anyone does have any additional questions, please just add them at the end, and then in the chat, just specify which speaker you'd like to answer your question there,
Sure.
and then we can direct questions accordingly. So again, thank you very much, Leighton.
I have
How's it
one
going?
quick. Yeah, for sure.
Sure.
So you said that you should start growing the second generation after like 2 growing seasons. So let's say you're doing a 1 and 3 year rotation. Would it be that 6 years then, or?
Yeah, that's right.
Okay, there you go.
So it's once you've grown a first generation hybrid, whether it's ours or whether it was maybe one of ours and one of somebody else's first generation, basically all of the first generation hybrids out there are the same genetic package. So if you've grown a first generation 2 times on that piece of land, that's when you should consider growing a second generation for the 3rd time. So hopefully, like I said, you're following up, being able to follow at least a 1 and 3 year rotation. So year 1 if you had canola, and then year 4 if you had canola, and then year 6 would be when you would, like you said, the 6th year would be when you would switch to the second generation.
So you're still recommending the switch to second generation in the 6th year, even if, let's say, I'm using, your product and then switching it to Bayer. Like, are you still saying that doesn't work?
That's correct, because first generation, basically, the first generation of genetics, the diversity in the genetics is not very large. The first generation, it can be said that they're exactly the same, no matter which company you're using. If it's first generation, you're not really doing a rotation of genetic sources by switching to a different company. So, and then once you get to second generation, again, there's not endless number of bullets or number of genetics that we can deploy. And we're not just, waiting for the 3rd, 4th, 5th, 6th generation. It's really quite limited. So really be careful and respect the rotation and respect the genetics, because they definitely can. I think Dr. Strelkov would attest to, the research suggests that after 2 cycles, you're probably starting to play with fire. You might get away with it. Look at your whole history, the holistic approach to this. If you're doing things by the book on every aspect, maybe there is another cycle that can go in from a third cycle for the first generation. But I would say, as a general rule, you should be thinking about the next generation.
Thank you.
You bet.
Okay, thanks, Leighton, very much for your presentation there. And now we will stem off into our next presenter here. So our next speaker is Dr. Steven Strelkov, who is a professor of plant pathology at the University of Alberta. He received a Bachelor's of Science from the University of Alberta, followed by a Master's of Science and a PhD degrees in plant pathology from the University of Manitoba. After a short stint at the NSERC visiting fellow with Agriculture and Ag Food Canada, Steven had accepted a faculty position at the University of Alberta, where his research is focused on disease of field crops, with an emphasis on Clubroot of canola. He serves as an editor-in-chief of the Canadian Journal of Plant Pathology. So now I'll hand it over to Steven for his presentation.
Okay, thank you very much. Hopefully everybody can hear me. Let's see if I can share my screen here. Let's see. Is that visible to everybody, I hope?
Yeah, looks good.
Okay. So yeah, so thank you for the invitation to speak at this virtual meeting. So I'll be talking a little. So Leighton and Aaron already did a very nice job providing a good overview of management and a little bit of the big picture. So I'm going to be focusing on my short talk a little bit on the occurrence or the distribution of Clubroot as of this year, and a little bit about pathotypes of Clubroot and their behavior on different resistance sources. So before I begin, so I'm Steve Strelkov, as just as Brett just mentioned, I'm a prof at the University of Alberta. Just wanted to mention Victor Manoli is a technician that's worked with us for a long time, and he usually does a lot of the survey and Clubroot work for us. We have a postdoc, Joanne Gu, who's been doing a lot of the epidemiological work. Keisha Holman is a master's student who's been doing a lot of the pathotyping. Xiaofeng Huang is my another professor, my colleague here at the University of Alberta. She's a prof of applied plant pathology. Mike Harding is a plant pathologist with Alberta Agriculture and Food, and we also work closely with him on Clubroot, as well as Greg Daniels, a technician in Mike Harding's lab. So it's really a team effort, and of course the counties and the municipal districts also are involved in some of this work. So this is just a brief overview of my presentation. I just want to start off with a little bit about the distribution or the occurrence of Clubroot in Alberta, and maybe touch a bit on prairie wide as well. Then talk a little bit about pathotypes of the Clubroot pathogens, sort of what are pathotypes and which pathotypes are we finding, and kind of finish off talking about the virulence of Clubroot pathotypes on Clubroot resistant canola. So going back in time for about a year, this is a map showing the distribution of Clubroot at the end of 2019, so last year's data. But I wanted to include this because we had the data from Saskatchewan there as well. So we have Alberta and Saskatchewan, and we're hoping to get some data from Saskatchewan this year so we can update this map. But what I hope you can see is that kind of the yellowish, reddish areas are where Clubroot is prevalent or has been found, and the darker kind of orangey red, the more Clubroot has been reported from there. So as of the end of last year, we had about 3,300, over 3,300 confirmed cases of Clubroot in Alberta. There were about, if I recall correctly, about 50 or 60 in Saskatchewan, and we were finding it's being found with increasing frequency as well in Saskatchewan and as well as in Manitoba and North Dakota. So which are not shown here. But I guess what my point here was that, you know, when Clubroot began about 15, 16 years ago, it was really just centered in really around the Edmonton region, like Sturgeon County, Leduc County, and most of the other regions, even within Alberta, were thought to be free of Clubroot. And over the years, you know, it's been continues to move into what we used to think of Clubroot peripheral areas, and now, you know, it goes all the way into Saskatchewan. So definitely, and I like the video that was shown at the beginning of this meeting, that, you know, Clubroot, even if it's not present in a region right now, likely over time it's going to probably start to increase and move into areas where it's not present now, and it certainly will. I guess we can talk about managing and slowing down Clubroot. I'm not sure we can really talk about preventing completely the establishment of Clubroot. This just shows focused on Alberta, and this is updated for 2020. So again, the intensity of the color reflects, you know, how many Clubroot cases have been identified. And as Aaron was mentioning, of course, the more surveying you do, the more likely you are to find Clubroot. So some counties in the Edmonton region, like Leduc, that's a lot of surveying, so they find a lot. Others where Clubroot is less prevalent, maybe do a bit less. But anyhow, in Alberta in 2020, we do a Alberta-wide survey together with so the University of Alberta, together with Mike Harding with Alberta Agriculture, and of course we touch base with all the municipalities as we do the survey. But we do quite a thorough survey, and we looked at about 600 fields in this year's survey, kind of spread out throughout Alberta, and we found 66 new cases in this survey. And in our survey, we look at the severity of the disease in each field. So we broadly categorize them as mildly infested, meaning that the index of disease severity is less than 10%. We talk about moderately infested fields where we have between 10 and 60% disease severity, and severely infested fields where the disease severity is greater than 60%. And what we found this year was consistent with previous years. We had about 39, or roughly 2/3 of the crops, were mildly infested. So the Clubroot is present, but it's not very severe. 24 were moderately infested. So, you know, varying levels of Clubroot, but certainly more than just a little bit. And we found 3 fields that were really severely infested, that had very severe clubroot. So on top of that, there were about 313 additional cases of clubroot found by the county and the municipal district surveys that they conducted on their own, and then they shared the results with us. So we had a total of 379 new confirmed clubroot infestations in 2020. And this included the first cases in Grand Prairie, Wheatland, and Smoky River. So just trying to show you what we're seeing is, clubroot got its start in central Alberta. It's moved all the way kind of to the end of the agricultural areas, here in the foothills and right to the Saskatchewan border. And it's now moving more into southern Alberta as well as into the Peace Country, the Peace River region of northwest Alberta. And so really it's basically becoming a province-wide problem or issue. And so something we all had to be aware of. So that's where we are with clubroot. So it's present throughout most of Alberta. It's becoming a bigger issue as well in Saskatchewan, Manitoba. So it is something we need to keep on top of. Now, one of the issues that can make clubroot more difficult to manage is that it consists of multiple strains or what we call pathotypes. And these pathotypes, they're all the same pathogen, it's all clubroot, but they differ in their ability to infect different hosts. So some may attack some varieties of canola, some may attack others, some may attack more, be more specialized to certain cabbages. Some strains may be, attack, rutabaga. So these strains differ kind of in what we would call their valence profiles, their ability to cause disease. And how we do pathotype testing, how we tell them apart is we actually will purify, and I made a little illustration here. We take spores of the pathogen. So let's say we get roots from a county or something. We extract the spores, and this is under very, very high magnification what the spores look like. This what hopefully you can see my little marker, this little gray, black and white image here. We purify the spores, and then we inoculate them, or we apply to roots of a whole bunch of host plants that are known as, in plant pathology, as differential set. So basically, based on which of the plants are resistant versus which of the ones are susceptible to a particular strain, we group the clubroot into a particular pathotype. And in Canada, we use a system called the Canadian Clubroot Differential Set for this purpose, also known as just the CCD set. And so what we found is that over the last, we started to pathotype, when clubroot was first found around 2004, 2005. But we've really seen a large increase in pathotypes since the advent of genetic resistance around 2009, 2010. What's happened is, as we introduced resistant sources to our field, we exerted pressure on the clubroot population to shift and to adapt. And so we brought new pathotypes that probably were already present in very, very, very low numbers, and they became more predominant, more common. And you can see there's 19 pathotypes that can overcome resistance in at least some clubroot resistant canola. And this is just more for your information. You just get an idea. The ones that are marked in pink here are the ones that break resistance. And it's quite a long list of pathotypes. And there's 17 pathotypes that do not overcome resistance. So we know of overall 36 different pathotypes that we've been able to identify from Canada. Now, this is a lot of pathotypes, and it's not realistic to expect companies, for instance, or breeders to try to breed for resistance against all of these pathotypes. So what they've done very recently is they focus their attention on those pathotypes that are more common. Because despite the very large number of pathotypes that we're talking about, really only a few are common or widely distributed. So most pathotypes, we have 36 pathotypes. Most have been found only in just a couple of fields, maybe in a group of fields. I don't know, for example, up in the Peace Country, there's only a relatively fewer pathotypes that are found commonly throughout most fields in let's say in Alberta. So this shows the prevalence of pathotypes, clubroot pathotypes, across Alberta, Saskatchewan, and Manitoba. And this is a large-scale trial done we did with a student called, who I mentioned at the beginning, Keisha Holman. And what we did is we pathotyped 166 isolates collected in 2017, 2018 from across the prairies. Most of them were from Alberta, so 146 of those 166 were from Alberta, but we also tested 10 from Manitoba and 10 from Saskatchewan because we didn't really know anything about the pathotypes there. And the pathotype with a little red star here means that they can break this first generation resistance. So what you can see in Alberta is pathotype 3A and 3D, which are both resistance breaking pathotypes. They're variants of an old pathotype called pathotype 3. They're by far the most common, particularly in fields with clubroot resistance issues. Pathotype 3H, which used to be the most common pathotype before genetic resistance was introduced, is probably the third most common. So it's still quite common, but it can usually be controlled very well by almost anything that is claimed to have clubroot resistance. So it's controlled by both first and second generation resistance. In Saskatchewan, we test a lot less samples, but you can see pathotype 3H is still predominant there. And there's another one, 5L, that also, but neither of them break resistance. So at least as of 2018, the last testing we did more thorough, there didn't seem to be any resistance breaking issues in Saskatchewan. Manitoba was kind of unique in the sense it was by far the most diverse in terms of pathotype composition. We found lots of pathotypes there that we didn't find anywhere else, including 3A. We did find out when they break resistance in one field, and a couple of fields had 3H, but then there were a number of other kind of unique pathotypes. So each province has a slightly different situation, but certainly 3A and 3D are the most important, and 3H is definitely still common, so we can't forget about that particular pathotype. So Leighton mentioned first versus second generation resistance, and I've also talked about it too in my presentation. So just want to touch a little bit on that. So the resistance in most clubroot resistant canola varieties that were introduced from 2009, when the first variety came out, to 2014, nearly all of it appeared to come from the same source, which was an oilseed rape, which is very similar to canola, winter oilseed rape from Europe called Mendel. And this resistance, the base of the resistance, is often referred to as first generation resistance because it was the first type of resistance that became available. And most infected clubroot resistant canola samples received and tested by us up to 2018 were virulent were strains of the pathogen that were virulent, could cause disease on first generation resistance. So that resistance began to be overcome or began to break in an increasing number of fields. By last year, we had over 300 fields that we knew had clubroot resistance issue. This year, we're still doing that analysis, but definitely over 70 new cases of potential resistance breaking. So of course, this was a big concern by all industry, given that resistance is our most effective and widely used method to manage clubroot. So since the emergence of these new P. brassicae or clubroot pathotypes, there's been an effort by many breeders and companies to develop canola with what I'm just generally calling here second or next generation resistance. And different companies call it different things, but basically it means something that they're carrying some resistance that's different than first generation resistance. So it's not the old Mendel derived resistance. I should say also that the base of the second generation resistance is generally not in the public domain. So it's proprietary information, so we don't necessarily know what occurs in different products from different companies, and they very well may differ across varieties. And this is similar to what, it's a little bit outdated, but similar to what Leighton showed, just clubroot resistant cultivars. This is from the Canola Council. I need to update the newer slide. But basically, there's a number of products out there that carry a resistant trait different from first generation or Mendel derived resistance. Now, it's very nice to have these new varieties as tools to manage clubroot, but we also have to remember that they are not magic bullets. Just like first generation resistance has been broken or overcome in some fields, second generation resistance can be placed at risk if we are not careful how we plant it, if we plant it too often in short rotations in fields that have clubroot. So for example, of 134 samples we received in 2019 that had potential resistance breakdown or erosion, 23 were samples or fields with second generation resistance. And this represented 6 different varieties with second generation resistance from different companies. So these strains are being tested right now with additional samples that we collected in 2020. So I told you in 2020, we found up to now 70 fields, and we're still analyzing the data. There's probably going to be more, but 70 fields or 70 canola crops where there appeared to be, that was club with resistance, but apparently there are strains of the pathogen that break resistance there. A number of these, at least 5 of them, are second generation resistance. So we want to test these as well. But what we've done with Keisha Holman, the grad student, is we've been trying to see how these, we basically have taken the strain of clubroot or the pathotypes of clubroot we're getting from the roots of canola with second generation resistance. We purify it, and we've taken a number of pathotypes and kind of compared how they behave on varieties that have second generation resistance. So what we see here in this graph, we have what we, if you look along sort of what we would call the y-axis, on the vertical axis, an index of disease as a percentage, so how severe the disease is. And we have these different bars, and each bar represents a different host variety. This one, ECDO2, is a highly resistant Brassica rapa from Europe that we often use what we call a resistant check. Typically nothing from Canada can infect this ECDO2. So we always include that just to make sure everything is working right. And you can see it had very little disease. ECDO5 is what we consider a susceptible check. As far as we know, this ECDO5 is susceptible to absolutely everything, every strain that we've ever tested. So we always just include it to make sure that the reaction worked, that everything is correct. Then we included a first generation resistance check just to see how that behaves. And as you can see, even though it's first generation resistance, it's quite susceptible when this is basically an average of the reaction to 10 different pathotypes obtained from second generation resistance. And you can see that this first generation resistance check is no longer very different than a susceptible check. And then we looked at 8 different canola cultivars or varieties from different companies which have some kind of second generation resistance. And what you can see is that in most cases, these pathotypes obtained from second generation resistance can cause some disease. In some cases, it's quite severe disease. In a few cases, they still perform quite well against this. Second generation resistance is performing quite well against these newest pathotypes. So this is just to show you, second generation resistance, it seems to be working in most fields, but we're already starting to see some strains of the pathogen that break both first generation and second generation resistance. So we have to be very, very careful how we deploy and how we use the resistance, because I think if we start to lose second generation resistance, we could be in a bit of trouble, because now the breeders and the company need to start to look for third generation resistance. And it's not necessarily always, there's resistance sources out there, for example, this ECDO2, but it's not always that easy to bring it into the canola varieties we need to grow here in Canada or want to grow. So we do have a proposal right now to look at this issue in a bit more detail, to see how these pathotypes are behaving on second generation resistance. But it's just something we need to keep in mind. So in terms of conclusions, because I think I'm close to, if not over my time, clubroot is now very widespread in Alberta. It's found, I think, in 2/3 of the counties or municipal districts that grow any canola in the province. And it's definitely increasing every year in what we formerly would consider clubroot peripheral areas. So and in those regions where it's moving into, it's good to keep on top of it before it becomes, they become severely infested. Next point is, while the clubroot pathogen consists of many pathotypes, there's only a few that are quite common. In Alberta, that's pathotype 3A, 3D, both of which are resistance breaking, and 3H, which is the old pathotype 3 strain that doesn't break resistance. First generation resistance is now overcome by numerous pathotypes in hundreds of fields. And second generation resistance has been developed against the new pathotypes, but it's not a silver bullet. We still have to take good care of it because we don't want to lose second generation resistance. Oops, sorry. And just finally to end, I'd like to thank Alberta Canola and the Alberta Agriculture Strategic Research and Development Program for funding a lot of this work, as well as SAS Canola and Manitoba Canola for some of the pathotyping of the isolates from those provinces. The Canola Council of Canada and the SCAP program that was mentioned earlier, as well as, of course, county and municipal district personnel, the ag fieldmen and the county workers and municipal district workers, because they really are partners in this work. Thank you, and I'm happy to take any questions if there are any.
Doesn't look like there any came up in the chat box, but
okay.
Just based on time too, I think we'll just hold those questions till the end, and then please put them into the chat if you have questions to direct to Stephen, and then we can get Dan here rolling on his presentation.
Sounds good. So
yeah.
Just unshare Thank you.
So yeah, we'd like to definitely thank Stephen for his presentation and the insight from the U of A there. It's really nice to have, and it's really nice to learn that edge. So
no problem.
So additionally, we'll segue into our next speaker here, who is Dan Stanton. He is a canola breeder with Corteva AgriScience. He grew up and lives in Redwater area and has been with Pioneer Hybrid / Corteva since 1999. He's been working on clubroot hybrid development since 2007 and has developed a number of clubroot resistant inbred parent plants and hybrids. So I'll let Dan take it away from here.
Okay, thank you. So what I want to talk about a little bit is how we develop these clubroot hybrids and what we think are important keys in terms of making sure that we maintain these genetics over time, because as Leighton and Dr. Strelkov have said, the resistant genetics are somewhat limited, and so protecting them through proactive management is very important. So as an outline, I want to talk about a little bit about our sustainability priorities, some information about clubroot breeding and how we develop resistant inbreds and hybrids, some clubroot dynamics information, and then a little bit about long-term management. So at Corteva AgriScience, we had some meetings and discussed this a lot because of what we know about the resistant genetics, what we know what's happening in the field, and set these as priorities for sustainability in clubroot. And the first priority is to ensure that clubroot genetics are grown. The reason why that is because if we get too high a spore load, then the resistance breaks down too quickly and we'll run out of resistance quickly. The second is to understand the risk in the region of CR1 breakdown. So, most clubroot hybrids are CR1, the same source used across the industry for the initial clubroot hybrids. And so if your region is at risk, then, Leighton talked about some of the tools that you can use to decide whether you should grow a second generation or not. And I think understanding the risk is important in that. The third is to understand the risk on your individual farm. So, whether it's just the things that you're doing or is it the things within the region, and I'll talk about that a little bit. And then the fourth is canola, crop rotation, and spore population reduction strategies to keep these resistant genetics lasting for a long time. So when we talk about clubroot resistance sources, plants and diseases have co-evolved for millennia. And so diseases change over time, becoming virulent, and the plant genetic resistance changes over time to match those clubroot strains. This has created a number of different resistance genes in the wild relatives of canola, as well as a number of different strains that Steve Strelkov talked about. So what we try to do is we try to cross to some of these sources, and the success of this crossing depends on how related that source of resistance is to canola, Brassica napus. And so when we talk about the Mendel source that everyone used at the beginning for clubroot resistance, that was a Brassica napus winter canola. And so we could cross it readily, it would set seed, And we had a high level of success in recovering that strong 3H resistance from that Mendel. As we move away from Brassica napus to other sources of resistance, now that we've finished with that easy source Mendel that everyone used, things become more difficult. And so in Brassica rapa, the Polish canola that we used to see in Canada a lot, there are a number of different resistance genes, and it will cross readily with Brassica napus. The problem is that Brassica rapa is a population, it's not self-compatible, and so you've got a whole bunch of different plants, some of them carrying different genes, and so often what happens is it'll have multiple resistance genes, and you maybe only recover one of them. It might be one of 2, one of 3, one of 4, but that allows you to remove more resistance into the Canadian cultivars. If we look at Brassica oleracea, it becomes very difficult to cross with. So that's like cabbages, broccoli, cauliflower, those type of things. When we do oleracea crosses, we have to do things like embryo rescue. It's very difficult to get seed out of it. The seed that we do recover has very little of the Brassica oleracea genetics in it, and so the chances of getting those resistance genes through are quite difficult. And so it becomes harder to access those sources of resistance. When we look at the top of the triangle, Brassica nigra, it doesn't share any chromosomes with Brassica napus, and so it becomes very difficult. And there was a study with Sinapis alba that showed that less than 1% of the Sinapis alba genome was recovered when they did these interspecific crosses. So let's say we're trying to get a Clubroot resistance gene out of something like Brassica nigra, it's very, very difficult because we'd have to do hundreds and hundreds of embryo rescues, recovering very small amounts of DNA, and chances of getting a Clubroot resistance gene through in that method is quite difficult. And so now that we've got the easy Mendel ones done, it becomes harder and harder to bring in new sources of resistance. Some of them are good and some of them have issues, and so we want to make sure that people are managing the Clubroot resistance genetic. So what happens when we do this development work? So what we do is we find a source of resistance. In this case, let's just say it's Brassica rapa. We need to cross it to Canadian elite parents. So these are parents that perform very well in Canada. They make up our hybrids that growers produce yearly. They have Canadian canola quality. They have the Canadian disease resistance package, and they have the Canadian maturity and adaptation to perform well in our environment. We'll cross it to this source, and then we'll have to repeatedly cross it to elite parents. So depending on how easily it's crossed, how well we can produce seed out of it, we'll go through several iterations of that crossing to bring it to something similar to a Canadian elite parent, but bring in that Clubroot resistance. What we do then is we go through something called double haploidy. So we culture the anthers, and we use those to create plantlets. We grow those out to be complete plants, and those are new varieties that we hope carry the Clubroot resistance and also carry those important characteristics of Canadian elite canola parents. From there, it sort of splits off into 3 different segments. So the first is Clubroot testing. We want to see if we've recovered some of those Clubroot genes that we had in our source originally. Is it giving us the full spectrum of resistance that the source had, or is it sort of limited in that regard? The next thing we'll do is field testing. So we need to make sure it's adapted to Canadian conditions. We make sure it's flowering, maturing at the right time, but also that it has the necessary characteristics in terms of oil, protein, glucosinolates, saturated fat to be a Canadian cultivar. The other thing we'll do is we'll develop mapping populations. So we'll cross it to a susceptible line, create a population where some are resistant, some are susceptible, and use that for genetic mapping. So once we know where that gene is located, we can use that information to help us in our breeding of future parents with that resistance. Once we've gone through all of that, it could be anywhere from 3 to 15 years, depending on what that source is. With the Clubroot Mendel napus, it took about 3 years to get through that process. With some of the cabbages and things like that, it's a much longer term process. Once we've gotten through that process, let's just say it's 5 years, then we start putting it into yield trials. So what we start with is we start with a few trials or a few locations, but many, many hybrids. And over the years, we whittle that down to a few hybrids at many, many locations. So as it goes through the process, year 6, year 7, year 8, we're selecting parents that are potential commercial hybrid parents. We're evaluating it for Clubroot in a number of different fields with different pathotypes to try to make sure it's broadly useful for growers. And then we put it into the private and public co-op testing. So that's the testing for registration purposes in Canada. By that point, we've gotten through there. We're at around 10 years of development time. And in the next year, we can do an initial launch with small volumes of seed. So if we have resistance breaking down and new pathotypes emerging, our pipeline has certain sources moving through it constantly, but if we have to develop a new source because it's a pathotype that hasn't been seen before and it starts becoming predominant, it could be a long period of time before we get a cultivar out. So with this, I'm going to go to those sustainability priorities. And so priority one, ensure clubroot resistant hybrids are grown. Clubroot establishes by movement of spores, and we've heard that from the other speakers. A small number of spores will get onto a field, and if it infects a susceptible host, it can multiply massively and establish a new area. So that leads to widespread infection in that field and further spread. Virulent strains spread in a very similar way to that. So, we might already be in a clubroot established area, but if you've got a virulent pathotype in, a field where you've got wind blowing or water runoff, that could spread to a neighboring field. If it's a generation 1 clubroot resistance and it's a new virulent spore load that resistance doesn't work on, then suddenly it's infecting a susceptible host. So it can move, in a similar manner to the initial clubroot infection. And so that's something to keep in mind as we're thinking about how to manage clubroot on our farms. The best method to keep clubroot off your field is to keep spore loads low. It's one of the keys of sustainability, and the best spore load is, it's 0 spores. If you can keep clubroot off your field in the first place, that's one of the best ways to manage clubroot. So if we look at different sources of resistance just here on the right-hand side of the screen, CR1 is that original Mendel resistance that everybody in the industry deployed starting in about 2009, and it's still present in most of the varieties today. Within Corteva, we've got 2 other different sources of resistance. So we've got CR2, which is the next generation clubroot. The genetics are not related to the CR1 Mendel source or the CR3 source that we have. And then CR3 is another next generation clubroot, and the genetics are not related to CR1 or CR2. Within the industry, there's a lot of companies that call their new resistant second generation. And so any hybrid with new or different resistance than CR1, they're sometimes stacks with CR1, but likely they're all different for each company because there's no simple, easy way to get that similar resistance that we got from Mendel, where we could all just take that winter canola, cross it to our Canadian stuff, and come out with new resistance. So on the bottom, it's a whole list of CR1, Pioneer and Brevant hybrids, and we've seen resistance as one of the best management methods, and so we've tried to put resistance into all the hybrids that we release. So this is priority 2. And so it's, what is the risk of CR1 breakdown in your region? Clubroot has been established in the region for several cycles. Within the areas east of Edmonton, there have been several virulent pathotypes identified, or fields with virulent pathotypes identified. And the other thing we have to think about is selection pressure. So if the spore population comes from canola crops that had CR1, then they have some selection and enrichment for other types. So not just on your farm. So if you're thinking about the spread of clubroot into new areas or within an area, often we need to think of how much selection pressure has been on it in the past. So if the grower that the clubroot spread from has already grown CR1 3 or 4 times, that spore population has been under that selection pressure for 3 or 4 times. And so it won't be the same as when resistance was initially deployed. It'll be a different population that has, more enhanced virulent types, such as 3A or 3D, or maybe some of the other more rare pathotypes. Within the region, east of Edmonton, rotations are often tighter than 1 in 3 or 1 in 4 needed for spore reduction. So, if we're having longer rotations, we get those spore loads down, there's less selection pressure on the hybrids that we put in the fields. If we have tighter rotations, that leads to higher spore loads and higher selection pressure. So we need to consider that in terms of risk of CR1 breakdown. A lot of the spread of virulent types came from that core Edmonton area region. And, we've seen a lot of virulent types coming out of that region. So that combined with the selection pressure of having CR1 in them that adds up to, a different spore mix than we might expect initially. And the other thing is managing within a farm. So, if you have a virulent type on one field, you have to watch field to field spread of virulent types, because you don't want them impacting your whole operation, especially if they're ones that are hard to control. So priority 3, so when we grow a CR hybrid, there are always strains present at a low level within that population. And some of those are virulent, and in response to being virulent on a hybrid, those low-level spores will increase. We expect that some of these are already elevated in fields and regions that have had clubroot for several cycles. And the other thing to consider is that some rare pathotypes may not be well controlled by any clubroot genetics. So at low levels, the clubroot resistant genetics often have a broader spectrum than at high spore loads. So if you have a really high spore load, it's more likely to be able to infect some of the new genetics. We also have to make sure that we control those rare virulent types by rotation of resistance and crop rotation. So at the bottom here, I have a bit of a diagram. You start with a field where you've got a mix of races. I'm calling the blue ones 3H and the yellow ones 3A. If I grow a 3H resistant CR1 hybrid, some of those ones that are virulent to it, those yellow stars, increase their populations. If I grow that same CR1 again, that 3A pathotype will get even larger and higher populations within the field. If we switch to something that's resistant to 3A, that can bring back those 3A spores back down. But if you've all been looking at the red stars, those are rare virulent pathotypes. So they start at a very low level, and they're growing and increasing in population, regardless of whether it was 3H or 3A. And so those rare types can grow and grow and grow. And because there's no resistance to them, eventually overwhelm your field. So let's say they're one of those rare pathotypes where it's really hard to develop resistance again. Suddenly there's no answer to the clubroot within your field. And basically you'll have that 5-bushel crop that Aaron Van Beers was pointing out on the susceptible canola. And so, canola would no longer be an option on that field. And if you're spreading it from field to field within your farm, that may not be an option on your farm at all. The other thing to consider is that, these may not have started on your farm. Someone might have had a few cycles of growing them in the first place. And so if they spread onto your farm, that's also an issue. So priority 4, rotation and spore load management for long-term management of clubroot. And so what we need to think about is that spore load management is key to clubroot management. Our goal is to minimize the spore loads for all these known pathotypes, as well as the unknown pathotypes that are present, as well as invading your farm from off-farm spreading sources. So if you take a 1 in 3 rotation with a 2-year break with no canola or no host, that reduces the spores by 90%. So, if you can increase your rotation beyond that on heavily infested land where you've had a breakdown or a shift in population that causes a huge amount of galling, huge spore load releases, then increasing that rotation can be a valuable tool. CR resistance is a relative term. As it breaks down, it shifts to the clubroot susceptible. So, if clubroot 1 is no longer working, then you have a susceptible host, and you're going to get those massive spore load increases. So our goal is first to lengthen our crop rotation, especially on land with high spore loads. And our second goal is to rotate clubroot resistance to prevent repeated increase in rare types. And so if you look at the diagram here, starting on the left-hand side, the first crop there, the darker green with the yellow and blue and red clubroot, you can see we're using a 3H resistant CM1, for example. Between that, we've got 2 years of alternative crops, whether it's a cereal or a pulse with, weeds and canola volunteers controlled. The next time we go into canola, we're switching to the yellow type, which is resistant to 3A. And so, in that 2-year break, we've decreased our spore load significantly. We're putting a different resistance in, so we're not building up any single pathotype in our field. But, we have grown 2 different crops that were not resistant to that red star. And so we're getting an increase in that red virulent